Welcome back. We're going to continue with Chapter 2. Now we're going to cover exposure assessment and applied science. All right, so what have we talked about so far? Because it might have been a little while since you listened to the first lecture. So we talked about the nature of contaminant chemicals or toxicants. And we started off with environmental science, and we looked at fate and transport of toxicants in the environment. You remember that? Yes, all of those aspects of environmental science. And then we went into toxicology, and that was the fate and transport of the toxicants in the body. So, how do humans become exposed? What kind of biological harm is produced? And we looked, of course, at those dose-response relationships. Oh, yes, you remember those, yes. And then we said that dose-response relationships were based on toxicology testing and bioassays, particularly, well, starting off with bacteria, moving on to rodents. Now we're going to talk about the applied science of exposure assessment, quantifying human exposure and contact with environmental hazards. And later on, this will apply to how to control exposure. And that's one of the major points, right? Trying to stop exposure to these toxicants that are dangerous, right? Okay, so this is completing the conceptual model of exposure. This is bringing it all together. So if you look in the book, you can there are a lot of those figures. They're in black and blue, and it shows the exposure pathway, and it sort of builds. It builds on all of the topics. So if you look at A, this is looking at the source of whatever toxicant we're trying to examine. So this could be an emission or some concentration of the toxicant in the environment, but this could be very hard to pinpoint if you're trying to look at a specific source. Sometimes this source can come from many different areas. It can be coming from the wind, from the water. It can be in many different products that you come in contact with. So what usually works is just looking at the concentration in a particular medium. So for example, now radon gas is everywhere, but we can look at a concentration of radon gas in soil, and that can be measured, or mercury in water, and there you go. All right, so from there, of course, from these sources, the toxicants go through fate and transport, right? And then they get into our environment. They concentrate in the environment. And there are two terms that we consider when looking at the concentration in the environment. There is the microenvironment, and that's looking at a particular site, just one site. So some place where you dwell, your home or office. For some of us, we might dwell more in the office than we do in the home. So that's the microenvironment, as opposed to the personal environment. Now, the personal environment would be sort of like there's sort of like a border around you that certain risk factors come into contact with you. So it surrounds you, and it's in many different places wherever you go. So maybe different places in the city. Okay, so it's no one particular place. You come into contact with it. Again, maybe it's a bottle of water that you buy here. When you get to your job, then there you're exposed to it again in computer components, and you get on the bus and it's there in the seats that you sit in, the vinyl seats. So this would be your personal environment. All right, so, and then from there, there's the route of exposure. So this can be ingestion, inhalation, or dermal contact. So when it comes to inhalation, well, let's go back to radon gas. Usually humans take in about 360 to 600 liters of air per hour. So it might be in that particular, and it would be in that air that you're taking in with inhalation. Children, by the way, because they have smaller lungs, it's true they have a smaller capacity, but they tend to be a little more active, and they tend to be outside, and they tend to take in more, even though each volume is a little less, their quantity is a little higher. And they sometimes kick up a cloud of toxicants. In the book, they call that the pig pen effect. If you've ever seen the cartoon Peanuts, where Pigpen's roaming around and there's just these little dust particles following him around. So it can be different, and we will address the difference between different groups like adults and children and risk. So, of course, with skin, that's dermal contact. Ingestion, now that could be incidental. So let's say you were swimming in a pool and you're not supposed to be drinking that water, but okay, you open your mouth and water gets in and then you do swallow. Or maybe there's a toxicant in the air and it gets on your lips. You know how there's just such a cloud sometimes of dust. Well, different things can be in that dust. And then once on your lips, you drink your lips or you take a swig of water and you then accidentally ingest it, or that's called incidental ingestion. Okay. And then there's just the darn right hand to mouth exposure. This means that you are bringing it to your mouth through smoking or eating or drinking, that sort of thing. So that would be, of course, now this is all the exposure pathway right there. By the way, don't buy a house with radon levels higher than about 4 picocuries per liter. So just a little thought here when it comes to radon. All right, so then we get to the toxicokinetic part. You remember that. So here you go from exposure to your absorbed dose. What is that? Okay, again, it's passed through the human envelope, so it gets in. It's the part that's absorbed. All right. And then you have the biologically effective dose, and that would be the effective dose is just the amount that reaches certain targets or sites throughout the body, and that's where the action occurs. And then from there, you can have changes in tissue structure or function, such as maybe DNA damage in your cells. And then finally to G, you have the possible health effect. So you put them all together. So you start off here, this is your exposure pathway, and then you end up through the toxicokinetic pathway till you finally get your health effect. All right. Now let's look at biomarkers of exposure. Sometimes this is also known as the internal dose, and that's what it's called. But this is a quantification of toxicants that are in the body due to an event exposure. So this, for the most part, includes your absorbed dose and the biological effective dose. Okay, moving back here. Absorbed dose, biological effective dose. Stay with me. There we go. All right. And this is a little different from biomarker of effect, just to sort of compare the two. So as a biomarker of effect is talking about the exact change in tissue structure or function. So this is clinical disease. Biomarker means, okay, this is sort of that evidence you've got something inside of you. The toxicant is there in the body due to an event exposure. All right. All right. So total exposure. Okay, for that, again, we look at the biomarker substances found that indicate its presence. This is found in well, it's not always found, but many times you can find it in urine or blood or hair, but not all chemicals are easily accessible as biomarkers. Sometimes you just don't see them, so it can be rather tricky. Okay. So the importance, again, of biomarkers when investigating exposure assessments, they do determine if a patient's symptoms are due to ingesting a toxic substance such as diesel fuel, or maybe those symptoms are due to secondhand smoke. You want to figure out which one it is. They help you identify sources of community contaminants by industrial pollutants. They're used in surveillance and identifying trends and exposures. So for example, if you were doing some biomonitoring And maybe you were able to show that exposure to brominated diphenyl esters, BPDE, it's a flame retardant. It was found in a lot of clothing and carpeting and furniture. And you have found and has dramatically increased in human breast milk over the past few decades. And actually, this is true. This is one of the things that biomonitoring has been able to do. So it's really important in surveillance and identifying trends. So, and then once we get this evidence, well, this will help in policy making, political policy making, saying maybe we need to restrict this. Oh, yes. And by the way, it worked. With this evidence, a lot of these flame retardant chemicals have been banned. So that's a good thing. All right. So again, when quantifying exposure, of course, we think about the dose makes the poison. Sometimes you can be exposed to lower amounts. We already talked about that. And you don't get any effect. So the dose makes the poison. But how do you always measure that? So how do you assess exposure? So we can quantitatively figure out the dose of the toxicant, sometimes by examining the exposure event. And, you know, it would be nice if you can just look at the biomarkers, but sometimes you don't have those. So in that case, you look at exposure. Sometimes you look at the exposure event. And to do this, you can measure the dose, the toxicants that are in the environment. Many times you model this through a computer. There was a quote that I thought was kind of funny that said that no model is accurate, but many of them can be quite useful. So computer modeling, you have to do a lot of there's a lot of information that goes into it, but it's very hard to sometimes relate that to what really is going on in natural situations. So, all right. And sometimes you can do an estimation by records and questionnaires. So all of these by exposure assessment. All right. How does this relate to our exposure pathway down here? All right. Well, again, one of the ways that we typically prefer to quantify exposure is by looking at biomarkers. And again, we're looking at toxicokinetics down here. And this is one way to do that. We can get a dose estimate based on this. However, what if you can't get a dose estimate down here downstream? And this could be perhaps due to, well, maybe the toxicant has a very short half-life. And radon, for example, has a very short half-life. You only have a few days to determine that it entered the tissue. Or sometimes it can be in tissue and you can't really access that. Or maybe it takes a very long time to get a health effect. So what we can do in that case is we can move upstream. Okay, so maybe we don't know the absorbed dose or the biological effective dose or change in tissue structure or possible health effect, but we suspect there's still something, there's still exposure and risk going on. Then we can go up here to the exposure pathway and look at all of this information upstream. But again, this is going to be a little bit different, and it will take some computer modeling usually. Oh, that wasn't supposed to happen again. So, sorry, we're going to look at this part. All right. So to translate an event of exposure into dose estimation using environmental monitoring, okay, well, sometimes what we can do is we can look at the microenvironment or the personal environment. Just as an example, as far as how to do that. Again, let's say we're looking for radon, and it's very hard to look at that in using the toxicokinetic part downstream. So we can look at exposure pathway upstream. So we can test soil and air around the house. Okay. And there's a lot of equipment out there. Sometimes this can be very, very expensive. Certain companies provide this. If we're talking about our personal environment, well, then you don't want to drag this thing around. It wouldn't fit in your purse or your wallet. So maybe you need to carry something around that looks like this. You can wear a personal monitoring device. And there are different devices out there to monitor the environment, of course, depending on the toxicant that you're looking for. So again, you can use this if you're looking at your micro environment, micro environment or your personal environment. So there you go. Now, how do we measure this? If you looked at the book, there's an awful lot of information and there's some calculations. I'm going to cover this. I'm not going to spend a lot of time on this because there just isn't a lot of time. But usually what we do is when we're looking at a particular chemical, a toxicant, we're looking at milligrams of the toxicant per kilogram of body weight per day that the individual is exposed. So again, you want to look at the quantity, the mass of the toxicant that we're looking at, and depending on your body weight, that's going to have a big effect, right? Uh, if you're a large person like I am, you have some fatty tissue, it can store there. So it's not going to the exposure risk is going to be much less in my in well, it's going to be the risk is going to be less, exposure risk is going to be less in someone like me rather than someone like a child. Okay. So, and of course, you're going to average that over time. So you might calculate how much per day that is. Other sources of exposure, well, if you want more information, you can take questionnaires, have people take diaries. One of the things that I won't go into the surrogate measure so much, but GIS, Geographical Information Systems, this is really big now that everybody has smartphones. And and there's just so many ways to assess particular environments and to analyze and interpret data in particular geographical areas. So it's a computerized system. You have special software, but what it allows you to visualize contamination, analyze and interpret the data very rapidly. And then using the computer, you can find relationships, patterns, and trends. So if there's some kind of cluster outbreak somewhere, you can hopefully analyze that and quickly find out where the source is. And this is a good example of a GIS map. And this is actually looking at a chemical plume in groundwater, and it can show private wells on nearby properties located throughout the entire area. So, and shows the areas at risk, which of course, again, that's one of the things we want to be able to do. Now, let's go into epidemiology. Now, there is a class that I will be teaching called epidemiology, and what this is a quantitative research method that is used. I'm not going to go into as much detail in this class as I will in the class called epidemiology. I'm just going to give you the greatest hits, okay? I know, you're welcome. So let's define epidemiology. It is a quantitative research method that studies the distribution of health and illness in human populations and links health outcomes to risk factors. So it really is, it studies the effect on human health from physical and chemical factors in the external environment. So, like I said, epidemiology studies the distribution and cause of health-related states. And we look at epidemiological information to give us knowledge to apply that in order to improve health and prevent illness in human populations. And so this really, when you think about what an epidemiologist does, think about what people from the CDC do, and they're always looking at, okay, what is the source? There's a problem. We want to find out what what the source, what's the risk, and we want to prevent it. We want to prevent this illness from occurring again. So it's there to improve health. And also, epidemiology does study the contrast between existing disease and what is expected. That's something else too. So those are risk factors. So environmental epidemiology examines potential health consequences from suspected exposures. This science draws on toxicology and exposure assessment. So everything that everything we've talked about, this is epidemiology. It draws all that information. So, for example, again, if you wanted to find out if cardiovascular disease could be caused by exposure to particulate contamination in the air. Okay, that's something that an epidemiologist goes out and they go find that answer. Usually you start with some assessment of the past or current or future exposures. They use a lot of computer models, like I said, to help predict future exposures. They design a testable hypothesis. So this agent has a particular health effect. And sometimes, though, if they can't do some kind of study like that, they might just do another type of study that tests people exposed versus people not exposed. And we're going to revisit that in just a little bit. There are challenges to epidemiology, however. Whenever you're doing an epidemiological study, it's hard to get a large sample size. So not every study size is as big as Flint, Michigan. Risk estimates tend to be rather low, so you can't find large exposures. And again, for statistical analysis, you need big numbers if you're going to find a relationship. Okay. Sometimes environmental factors take a long time to cause disease. They hide, they can be latent. We talked about cancer and how, all right, it usually takes several different steps to get there. We could also talk about something like Alzheimer's and multiple sclerosis. We know that there's an awful lot of steps that are involved in that. There could be multiple causes of the disease. You come in contact with lots of different factors. You could have synergism. Remember, the two factors coming together have a stronger impact than if they acted independently. So 1 + 2 = 4 kind of thing. And sometimes even if you get some answers, they really don't quite tell you for sure, black or white, yes or no. There could be something in the gray zone. So these are epidemiological challenges that are faced by people from like the CDC. So when talking about key measures of health status of populations, there are some terms that you need to know and terms that are good to know for the test. Some of these I assume you already know, but we're going to cover them anyway. When we talk about mortality, that is the incidence of death. Morbidity, that is a diseased or morbid state. So basically, you have the disease, okay? Different than mortality. Mortality means that you have passed, you have died. Disability, a substantial limitation on a major age-appropriate life activity, okay? And the book didn't have it in this order, but I wanted to call, even though mortality talks about the incidence of death, I wanted to compare incidence and prevalence. Sometimes this is hard for students to be able to tell apart. So first of all, incidence. Incidence is new cases. I have this highlighted in red. So it's new cases of disease during a given period of time. Prevalence is a proportion of the population with disease at given points in time. So the best way to demonstrate this, okay, I'm going to go to a bigger illustration. I found this online, whoever did this illustration, and I have the blog down there. They did a very good job just looking at trying to explain this. So let's say, so here we have prevalence. This means that, all right, these are all the cases of disease. So each little circle represents one case. So this is sort of like your baseline, okay? This is how common it is, how prevalent it is. So incidence means new cases during a particular exact time. Now, these incidents, these new cases, then join the prevalence individuals, right? So then they increase the prevalence and beyond the baseline. So here's the baseline. Incidence is new cases. So these new guys join the old guys and they all become one big population. All right. So prevalence, once your prevalence is again just how many are there. Now, sometimes you have cures and deaths, and you get decreased prevalence as a result of that. So this just puts it together, and I sort of like that relationship. I like the way that this illustration puts these that they describe these two concepts. Okay. So now let's look at descriptive epidemiology studies. So descriptive epidemiology, that must mean that it describes epidemiology, and it does in a particular way. Again, it refers to different kinds of studies in epidemiology. We'll talk about a few others later on, but it describes patterns of disease in populations. It's looking at who, what, when, and where. All right, that sounds pretty reasonable. When little is known about the disease, that's usually when we go into a descriptive epidemiology study. Typically, we need to find some pre-existing data. We need to find some information to launch this particular study. We look at a lot of case studies or case series. So these are noteworthy sets of cases that we know have some good evidence, some good data. Okay, we can do a lot of surveillance in a descriptive epidemiology study, go out and track and compare disease rates across places, diseases, and times. Usually surveillance does require biomonitoring, using all of the biomonitoring tools and not just the apparati, but also looking at documented rates of exposure or looking at disease in populations. And the CDC, whenever you think of the CDC, I always think of them as a bunch of descriptive epidemiologists because they're always doing this. There's some cluster of disease, there's some outbreak, and this is what they're doing. They're like, okay, we don't know what's going on. We just know some people are coming down with certain signs and symptoms. We're going to figure out what's going on here, look at a few case studies. We're going to track it down. Now, if you've ever seen the movie Contagion, in the movie Contagion, it has a lot of epidemiologists doing a lot of descriptive epidemiology, at least in the beginning. Okay, and then they move on to different types of studies. Okay, so I want to just really quickly talk about compare descriptive versus analytic. And within analytic epidemiology, there's even a few other specific types of analytic epidemiology. But just to compare the two, descriptive, again, we use surveillance. We're looking around. We're looking at who, what, when, and where. We use it when little is known about the disease, and it usually illustrates potential associations. When we get into analytical epidemiological studies, pretty much we're looking at why and how. Because we already know some certain characteristics or aspects of the disease. So we've already figured a lot out. We already have some other evidence about it. Usually it evaluates the cause of associations, and that's where we'll come back to this. All right, now, we always have to look at epidemiology has a lot of math involved. I'm really going to keep it very minimal for you in this particular class. At least for now, we might bring in some other types of, we'll probably have to bring in some other math later on. But for now, for this particular chapter, I'm going to keep it short and brief. Yes, you're welcome. So sometimes we look for, when we're trying to calculate incidence, we look at just a crude incidence rate, some kind of rate. And again, crude means just, it just means straightforward. It's not looking at many details, okay? This is most commonly used at the very beginning of disease surveillance. Now, when doing a incidence rate, you always have a numerator over or you have the numerator over the denominator, right? So the numerator is, this is going to be really obvious. This is going to be the number of new cases that in the population. So you start to see little outbreaks, right? And there's this person and that person, and they're starting to get signs and symptoms. So that's an easy one to calculate. The denominator is a little harder to calculate. So the pros always look at the denominator. That's always the harder one to determine in a question, because now you have to look at everybody at risk. So persons and years at risk. That's the tougher one. So you have to ask that question, well, all right, so this many people came to the hospital. These new cases appeared in the hospital at this particular time. But is everybody at risk? If we want to do a rate, if we want to find an crude incidence rate, we have to figure out, okay, who is at risk? So it's a quick calculation. You do the numerator over the denominator, 300 new cases in a population of over 100,000 over a 10-year period. That's going to give you a crude incidence rate of about 0.0003 per year. Okay, because instead of add you want to make it per year instead of over a 10-year period. So if you did that calculation, 300 over 100, or excuse me, 300 over 100,000, and that would give you 0.003, and then over a 10-year period, divide by 10, you get 0.0003 per year. That's just a crude incidence. Okay. All right. The problem with doing a crude incidence rate is that, well, many diseases occur at different rates in different types of groups. So, for example, if we were measuring in a population a crude rate, incidence rate of breast cancer, and we just, let's say we wanted to find out the crude incidence rate of breast cancer in Omaha, Nebraska, and we could come up with that number. But that's kind of silly, isn't it? Because now, even though some males do get breast cancer, primarily we find this in females. So it doesn't account for that. We don't have we don't define, okay, which part of the population is male, which part is female. Also, you have to look at age because, of course, breast cancer is more common in females as they get older. Actually, all cancers are more common as you get older. So the crude rates for different populations or same populations at different times, they're just not comparable. So we have to do something to standardize that. Okay, that's the solution. So we could do an age standardization, or we could do a gender standardization. I just want to show you just one example that I found. Okay, so for example, let's just do the age standardization for cancer. Okay, and we wanted to look at the number of deaths due to cancer. And we looked at Florida and Alaska. And if we did the crude evaluation, okay, the crude incidence rate, then in Florida, you would have 131,902 over 12,340,000 individuals. So the crude mortality rate is 1,069, and that would be your rate, as opposed to Alaska, okay, where you have, again, the rate is considerably lower. So does this mean, wow, I'm just going to go to Alaska because I won't get, I won't get cancer and die because they have such a low mortality rate? But if you look at this, though, the numbers, they're quite different. And Florida, it's just looking at numbers of in the population. It's not looking at age. And of course, if you're older, are you going to, where are you going to go if you're older or during the winter? Are you going to head for Alaska? No, most people go to Florida, and Florida politically makes it very attractive. So Florida is full is has a high older population there. So of course their crude mortality rate is going to be much higher than Alaska because their age distribution is higher. So okay, you can't really do that. So you need a standardized rate, and this allows you to do this. So you can look at age-specific mortality rates in Florida and an age-specific mortality rate in Alaska. And there you go. So this is a little more, this is going to help you calculate according to age. And if you look at the age groups in the distribution here, okay, they're pretty much the same, and this is just hypothetical, but you still have the same mortality rates in Florida as well as Alaska. But the crude rate, oh, sorry, this was cut off. I'm sorry about that. So, but the crude rate would be very different. Sorry about that. All right, so let's go back, and again, we're going to compare descriptive versus analytical epidemiological studies. So again, descriptive study designs, okay, they use surveillance and illustrates potential associations, and they're looking at all of the factors. They're not quite sure what the cause of the problem. They're unaware of what's going on, so they don't really know a lot about the disease. As opposed to if you go to analytical, and okay, then we know a little bit more. And within the analytical epidemiological study design, we have observational and experimental study design. So within analytical, you have these two others. So again, all of these, both observational and experimental analytical study designs, look at risk factors and health outcomes. Basically, it's why and how, because again, we've already ascertained several aspects of the disease. We already pretty much know a lot about it. We're still trying to, again, we're evaluating the cause of the associations. So the difference between the observational and the experimental is the observational is at that point when you're looking at a population, you are not manipulating exposure. You're just looking at a particular group. You're observing. Uh, you're not changing any variables. You're just taking data, okay? As opposed to an experimental study design. And if you remember your general biology class, you probably remember that in a good experimental study, you probably want to have, some controls in there. And usually certain things are manipulated. You usually have two different groups to compare. So the experimental study design uses different exposures and treatment groups to compare, like placebos, exposed, unexposed, that sort of thing. And you can look at this both at a group level or an individual level. I found this online. I thought this was a nice way to just break it down. So when we're looking at epidemiological studies, you can go descriptive, you use surveillance, you kind of qualitative. If you do analytical, you have experimental and observational analysis. So again, experimental, you're going to probably have control groups, you're going to have some manipulation of your experimental groups. Observational, you will not. You're just observing. So little more about that in just a little bit. Okay. So under the analytical study designs, again, you have the observational part of study design. And even within that group, you have what is called the group level observational study. And for that type of study, you're looking at a group, but you're just documenting patterns and rates. And that's all you're doing. You're not even necessarily looking between for an association between characteristics of communities or groups. You're just collecting data. It doesn't even try to explain patterns or identity factors associated with health outcomes. Um, Also, you have to be careful because it does sometimes it's not very objective and it's very subject to bias. So with this, and it's not really designed to test a hypothesis. So, you know how you come up with a hypothesis, I think this is happening because of this. It's not really designed to test a hypothesis. It's really designed to generate a hypothesis to help you look at just looking at these documents. You might think that there might be, yeah, that there's a relationship. But still, community level factors, they are still important. Now, as opposed to an individual level observational study design. Now, with an individual level study design, you have two sets of individuals. This is what I'm more familiar with. I like being able to compare two sets of groups. For example, compare one group with disease and one without disease. So one with exposure, one without exposure. So this allows you to have some comparisons. And again, this is what I'm more familiar with. This is designed to test hypothesized associations between exposure and outcome. So this is, again, all part of analytical epidemiology. So within the analytical study design, sometimes you hear the word cohort study, and that deals with a group of subjects that are all selected according to exposure status. So for example, a cohort group would be people working in a research facility. That would be where I came from. So we all had, let's say within our group, we were looking at uh, perhaps exposure to chemicals that caused cancer, that sort of thing. So you would do a comparison on disease status and just really looking at, okay, if you are exposed, are you more likely to be diseased? So who is exposed to this particular chemical? Are they more predisposed to being diseased? Now with this, you can look at, you could look retrospectively. You can say, okay, these people were exposed to this chemical. They were more likely to be diseased. And you can take that data, retro being in the past. But in addition to that, it's also possible to continue studying this cohort group and then take intervention steps and keep these groups away from these particular chemicals and see if the rate of exposure or just see if the health effects are reduced and the rates decrease. So that's one of the nice things about cohort studies. Now, when you look at case control studies, that's a little differently. You're basically selecting people according to their disease status. So everyone you choose, okay, they're either they have a disease or they don't. They are, for example, maybe they are smokers or they're not, and you look for lung cancer. Okay, so you're looking at that's case control study groups. So you get a group of people. Have you ever smoked? Yes, you have. You're one group, and then you've never smoked. You're another group, and then we compare lung cancer rates. So again, our case is more likely than controls to have been exposed. So if you are smoking cigarettes and the group that smokes cigarettes does not have a higher rate of lung cancer than the control, the people who never smoke, well then you really can't see it. It doesn't look like there's a link between that. All right. Now, when evaluating individual level studies, you have to look at if the findings are statistically significant. And if you've had any kind of statistics, you know that this is important. You can come up with all kinds of numbers, but are they significant? Sometimes you get numbers and they're due simply to chance. So you're pretty much up there 50-50. Also, you have to look to see if there's any bias. So how are the test subjects selected? I have a little example of that. So you want to make sure there's no bias. Your test subjects are selected either, I mean, you, you, uh, you don't want to have any bias when selecting your test subjects. Okay, plain and simple. Um, Also, you might find that your data has some confounding factors. So your finding is due to a confounding factor. What does that mean? So, for example, if you had like a you're looking at uh, uh, body mass index and does it cause heart attacks? So you're looking at people who have a high BMI, and okay, yes, they have an increased rate of heart attack. However, turns out that individuals with higher BMIs, they tend to be older. Well, we also know that age is a risk for heart attack. So you have these two confounding factors. So you really you would have to look a little deeper and redesign your study. How about effect modification? Maybe you have something going on like synergism or antagonism going on. So you're trying to find a link. Does cigarette smoking cause lung cancer? But then you have people who are smoking cigarettes and they have lung cancer, but they also drive a diesel engine or something like that. So, okay, the two of those together are going to give you a much higher rate, right? So that would be a synergistic effect, if you remember that. Okay. Okay. I wanted, when it comes to bias, let's go back to, you have to be careful with your test subjects and you want to select them for with bias, and you want to make sure that everything is not due to chance, and a few other things. But I wanted to show you this. Because I teach microbiology, I also teach immunology, and we have to talk about vaccinations. And okay, so you probably know there's a lot of people that feel very strongly about vaccines, and they believe that vaccines cause problems from multiple sclerosis to autism. You have heard that, right? Well, scientists have to be really careful about linking two factors and saying, okay, this causes that. You can't jump to conclusions when you're a scientist. And when you're doing epidemiological studies, you certainly cannot. So, Andrew Wakefield, now he was a British gastroenterologist, and he was a lead author in an article in the Lancet. Now, the Lancet, if you don't know, is sort of like the Super Bowl for researchers. When you are published in the Lancet, well, you are considered an opinion leader. Now, during this time, there was an awful lot of interest in the link between vaccines and other problems like autism. And people were emotional about this because the rates of autism were climbing and people wanted an answer. Okay, well, this gastroenterologist was going to provide that answer. So he looked at a study. His group of he looked at 12 random children, and with his work that he published in the Lancet, he connected measles to a component in autism. All right, so have you had statistics yet? All right, is there a problem with the number 12 for you? Are you having problems with that? Yes, you probably should have a problem with that. The word random, you probably don't know you should have a problem with that one as well. So remember, studies are not supposed to be biased. Unfortunately, lots of problems occurred. Now, we won't even go into the fact that a lot of the data was fraudulent. He outright changed the data. The Lancet did have to retract the article, by the way. So Andrew Wakefield received money from a law firm representing parents involved in vaccine lawsuits. He also personally took out a patent for an alternative measles vaccine. Whoops. And he did alter the records, as I said. By the way, the random children were referred by the law firm that was, well, basically, uh, 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 they were involved in the lawsuit. Hmm. Little side note too. He lost his license. Oh, also he paid children at his son's birthday party to participate in blood drawing for the experiment, but that's an ethical question that doesn't really enter into epidemiology. But of course, we can have a problem here. You know, sometimes people look at a study and they love to look at data that supports their view. They look at numbers and they want to see things that aren't there. But an epidemiologist, a scientist can't do that. Unfortunately, you know, is he the main villain? No, the Lancet should have caught that. That's horrible. Most of my students, if they just looked at the data, could probably see that there was a problem here. But people get emotional and sometimes they don't look at the information in front of them and the facts, okay? And the media, of course, was a big problem with this. Okay. So, one of the things you can look at, there's something called the Bradford Hills criteria for use in evaluating causation, sorry. And this allows you to see if, okay, the information that you have uh, that provides sort of a link between perhaps a toxicant and a health effect. So you think, okay, the data says there's causation here, but is there really an association, a causal association? Well, hopefully this will help you, and scientists, epidemiologists use this frequently. So this just says, okay, let's look at the strength of the association. Let's look at the numbers. If you are, you know, 1 time more likely when exposed to this toxicant to develop this health effect, not so good. But if you're 10 times more likely, okay, that's stronger. Consistency of findings with every experiment, wherever it is. All right, you keep getting the same outcome, so it looks like there's causation here. That's pretty good. Looks like a good link here. Appropriate temporal relationship. What does that mean? So that means that you didn't get the disease before exposure to the risk factor. Okay, so you didn't have the heart attack and then become exposed to the toxicant. So you want to make sure that there is a separation there between the risk factor and when the disease occurred. So also, of course, look at a dose-response relationship. So this is something biological. And of course, the risk should increase with increasing exposure. And biologically, is it plausible? So if you look at at biological mechanisms and you see this chemical and you say, okay, what this usually does is it binds to an essential enzyme in the mitochondrion, which is the powerhouse of the cell. And okay, so we can see where this particular chemical would cause a problem, not just in the cell, but in tissue and tissue system, or in organs and organ system. So is it biologically plausible? This is what you really need to do to evaluate causation. Okay. All right. Now we get to talk briefly about infectious disease epidemiology. All right, more about this a little bit later. Can you hear that my voice has gone up a few octaves? Well, it's infectious diseases. There are many methods that are employed to model transmission of certain infectious diseases. And I was just listening to an investigator from Great Britain, and they were trying to be able to predict when the next pandemic would occur. It's very interesting. So you would have to use again, you would use math and computers, and you would have to figure out how everything is transmitted and how particular infectious diseases act. But you know, the transmission of diseases are affected by many different features. Okay, so first of all, what's the pathogen? What is it that's generating disease? So, you know, certain organisms make toxins, and some of these toxins are extremely potent or virulent. Virulence is strength, strong. Okay, so like Clostridium botulinum produces the botulism toxin. It doesn't take very many of the little toxin particles to kill a cell. Bordetella pertussis causing whooping cough, nasty little toxin. So some pathogens have the genes to make these toxins, and that is going to affect how well it does in transmission. How is it transmitted? Okay, that's something else to look at. So what spreads the particular pathogen? So when we were looking at, okay, if you're looking at Lyme disease, that's being spread by ticks. If you're looking at things like malaria, it's being spread by mosquitoes. Bird flu, well, then you have it's being spread by migrating birds. So if you have, well, here's a good example. As migrating birds can travel over a continent in days, okay, so something like bird flu could be transmitted very quickly, very rapidly, and transmitted very easily. Something like African sleeping sickness that is transmitted by a tsetse fly, because of where it lives, it's an arid area, there's not a lot of these little tsetse flies to transmit disease, although it's very, very nasty. But transmission is a little inhibited because there's not a lot of vectors because the environment's kind of nasty. You also want to look at the individual person. I always tell my microbiology students, so why do we get sick? Well, usually it has to do with how virulent your pathogen is and how good your immune system is. If you have a strong immune system, and this takes into account many, many things. So your skin is intact, your barrier envelope is intact, you're eating well. That's going to feed your immune cells, which are like your soldiers. So you want to feed them well, give them what they need. Okay, maybe you've had a vaccine or previous exposure and you have memory cells. All of this is going to determine whether or not something is going to be transmitted to you. And of course, there's other physical characteristics that can play on your susceptibility to transmission and your social environment, that sort of thing. So as I said, if you lived in Africa in a very arid place, not a lot of water there, not a lot of tsetse flies. Again, certain areas, there's better hygiene. You have good water resources, that sort of thing. There's just an awful lot. If you live out in the country, you're isolated from people in the city if a disease breaks out. So there's a lot of different physical and social and environmental factors that come into play when we transmit infectious diseases. Okay, some terminology when we talk about infectious diseases in populations. First of all, endemic. Endemic means this is predicted. We can say, yeah, we have our rate of colds in the winter is always set. So we would say the common cold is endemic to Omaha, Nebraska. It is common, typically present at low to moderate levels in a population. It is what is predicted. Okay. Epidemic means that you have an occurrence of the disease at a higher rate than predicted in a population. So it's higher than predicted. Okay, so you have an outbreak. We always predict, that there's going to be little cases of influenza throughout the year, even in the winter, but every once in a while you have a higher rate than predicted, and then we call that an epidemic. Now, when you have an epidemic that goes global, you have rates higher than predicted, that is a pandemic. HIV and AIDS is still at a pandemic rate, meaning that, well, we know it's out there and we predict that, okay, in a particular amount of time, we're going to have this rate, but it keeps always going above that expected rate. So, and it's global, it's all over the world. So HIV and AIDS is a pandemic, a global epidemic. Okay. Animal epidemiology. Again, this is going to come back a little bit later on. Animals are sentinels for human disease. So, for example, we could look at mad cow disease, which is caused actually by a strand of amino acids, it's a protein called prions. And we saw that something was going on. We never really even started to study this until a bunch of cows in Great Britain started to act very strangely and fall, and we call those downer cows. And because of that, we were able to isolate these weird proteins in the cow brains. And it turns out that this is transmissible. How did the cows in Great Britain get it? Oh, I'm so glad you asked. Turns out that, well, you know, when you get a cow and you eat the best parts of the cow, right? So you take a little bit of the hindquarters, and that's where the steak is. What do you do with what's left? Like the brain, the central nervous system. Well, what they typically did was they would grind up what was left of the cow, and then they would heat it and make sort of kibbles and bits, right, for protein. And they would feed it back to the cows. Now, cows, of course, you know, they're not supposed to be eating cow. They're herbivores completely. But that's not the case. What they were doing is they were eating the brain material of infected cows, and it kept getting recycled in the community. So then it'd get into a new cow or herd, a new population. They would come down with they would get the prions, and so they were infected, but the meat was going out, and then their central nervous system was being ground up into cow parts. The scariest thing about that is, of course, they cook that pretty high. Think about what goes into Purina dog chow or cat chow. This is what was going into the cow feed. And so you cook this at high temperatures. It doesn't destroy prions. Prions are very hard to destroy. But animals, because of the cows, it gave us sort of a heads up of what's going on. One other thing to mention, the Black Death, the plague, one of my favorite topics. You could always predict that the plague was on its way because you would see a lot of dead cats, especially, but then also the rats and dogs would also die as well. Um, Speaking of animals, though, they do work as surrogates for human research subjects. And of course, we already talked about that when we were talking about rodents and bioassays. So it's very useful in order to be able to try to mimic what goes on in humans and human populations. I'm not going to go into that in any detail, but it's something that I might bring up a little bit later on, okay? All right, so next week, community-based participatory research. Ah, all right. You have a great week. Bye now. Oh no.